you for the introduction. Unfortunately, I'm not a single cell person. In fact, you could probably put me out of a job. My name is Irene Unterman. I'm an MD PhD student from the Hebrew University, and I'll be presenting CelFiE-ISH. It's a deconvolution tool for whole genome bisulfite sequencing. So all cells in our body have the same DNA. DNA methylation changes between cell types and allows for the diversity in cell form and function. Cell type-specific methylation patterns are established during embryogenesis and are actively maintained. DNA methylation changes have been reported with age, smoking, and cancer. But to truly study the role of DNA methylation alterations in human diseases, we must first account for the strong cell type-specific signals. Now, deconvolution is the problem of estimating cell type proportions from bulk methylation sequencing. I'll focus on reference-based methods where we know the different cell types and their respective methylation patterns, and aim to measure how much each of them contributes to the bulk mixture. And this is the first step towards correcting for cell type composition in order to find disease-specific signals. Now, the methylation sequencing technology defines the resolution of information that we have to work with. Many deconvolution algorithms have been developed for methylation arrays, where we only know the propulsion of methylation at several CpG sites across the genome. Now, whole genome bisulfite sequencing is becoming more popular, and there we have actual sequencing of short 200 to 400 base pair fragments. Now we know not only the proportion of methylation, but the actual methylation pattern. And several CpG sites can exist on a single molecule. As we move towards sequencing gradually into longer and longer fragments, the algorithms must also be adjusted in order to take advantage of the full breadth of information that is available. I'll show you how 4 different deconvolution algorithms treat the same whole genome bisulfite sequencing data. So here we have a few molecules from whole genome bisulfite sequencing. Full circles are methylated CpG sites, and empty circles are unmethylated CpG sites. And each row represents one molecule. What we know came originally from one cell. So this first algorithm was published by Krista Caggiano from the Zeitlin Group in 2021. And what it does is it looks at every CpG site across the genome and counts how many methylated CpGs it sees out of how many total. So 1 out of 2 for the first one, 1 out of 2 for the second one, and so forth. And as a result, you completely lose the within-read information, the fact that this entire row has one cell type of origin. A perpendicular approach is UXM. This algorithm looks at molecules and counts how many methylated CpG sites out of how many total exist per molecule. And if the molecule is mostly methylated, it is called M. If it is mostly unmethylated, it is called U, and short or ambiguous reads are called X and then discarded. This was published by the Kaplan Group in their 2023 Nature paper. And here you completely ignore the spatial aspect that CelFiE used. So the first of my algorithms is called epistate, and we can think of it as an extension of UXM, a softening of sort. The unmethylated and methylated states are defined per position. And instead of hard assignment, I use probability. This way, we can quantify uncertainty and include the short or ambiguous reads that UXM throws out. The final algorithm that is also mine, called CelFiE-ISH, is an extension of CelFiE with the added constraint that every molecule must only have one cell type of origin. So it just goes molecule by molecule, cell type by cell type, and calculates the probability of each molecule originating from that cell type. We have 4 different algorithms, and we wanted to compare them. We started with simulations. First, by reproducing the simulations from the CelFiE paper, where you have 25 simulated cell types, all mixed together at linearly increasing proportions. So the true proportions are the red circles. If you were to sum up all the red dots, you would get 1. And the gray boxes are the deconvolution estimates. So the closer the boxes to the red dot, the better the deconvolution. And to give a number to the overall deconvolution of all cell types together, we have the root mean square error. Then we added a twist. We added multiple CPG sites on a single molecule. So since this is the CelFiE underlying model, we can ignore epistate and UXM, which are looking at things completely differently, and focus on the blue and the orange lines below. So we have error on the y-axis, lower is better. At a read length of 1, and by read length I only mean number of CPG sites on a molecule, since I'm not looking at the DNA sequence, we can see that CelFiE and CelFiE-ISH are exactly the same. However, as you increase the CPG content or the read length, CelFiE cannot use that information, so it stays the same, while CelFiE-ISH keeps improving and the error goes down. Now here we have a second simulation, now with the UXM or epistate generative models, where every read can be either mostly methylated or mostly unmethylated. And we have a read length of 5. And the one thing I wanted to show here is that for the exact same data, the models that are able to account for within read information can deconvolute pretty well, while CelFiE, that doesn't take this sort of information into account, is not able to deconvolute this sample at all. So we saw very different behaviors for different simulations, and we wanted to see what happens in real human data. So we were very fortunate to have access to the Huji Atlas, which contains over 30 human cell types, pure fact-sorted cell types. And here we cannot control the CPG content. It is what it is, but we can control how much we sample. So we made in silico mixtures of the cell types at different proportions. And we varied the overall coverage from 30x, which is the natural coverage in this data set, all the way down to 0.1x. And again, the y-axis is error, so lower is better. And you can see that across all the different coverages, CelFiE-ISH had the lowest deconvolution error. Now, the last result that I want to highlight is the sensitivity of the algorithm. Here we try to generate a cell-free DNA-like mixture with a majority of lymphocytes and hepatocytes, with all other cell types set to 0. See? And one cell type that is a small spike-in. In this case, it's cell type number 26, which is cardiomyocytes. And then we took just the cardiomyocyte component and compared it against mixtures that look exactly the same but don't have this component. Okay, so let's start from the right, where we have a spike-in of 0.3%, and all the different deconvolution algorithms are able to tell apart between the spike-in and the null mixture, except for epistate. And as you go down all the way to 0.03% spike-in, that's a proportion of 0.0003, only CelFiE-ISH is able to distinguish the spike-in from the null mixture. But another thing that really stands out is that CelFiE-ISH is overestimating both the spike-in and the null component. There is a difference, but you can see they're both really high above the blue line, which is indicating the true proportion. To investigate this effect, we increased the coverage, which was 5x here, all the way up to 100x, and we're looking at the input cardiomyocyte proportion, percent, versus the estimated percent. And the first thing that we see is that UXM, all the way to the right, has the least amount of systemic bias. It is closest to the diagonal line. While CelFiE-ISH is indeed overestimating the rare component. However, CelFiE-ISH has a dose response. It's not a linear relationship, but you can see that there is a clear difference between null, which is the orange to the left, and even the 0.03% spike in, while in UXM, there is no difference between the two. So to recap, within read information or multiple CpG sites on a single whole genome by sulfide sequencing molecule can improve deconvolution, especially when the coverage is low. It can improve the overall accuracy and also binary detection of rare cell types. As more technologies for native DNA sequencing, which is very long and scarce, become more common, this information will become essential. As a future direction, once you have the best possible deconvolution, you can use that information to distinguish for differentially methylated regions, which of them are caused by contamination and which are truly associated with disease states. And with that, I would like to thank you for listening. I would like to thank my advisors, Professor Berman and Professor Glaser, as well as our collaborators. This work is just out in Genome Biology, and there is a QR code to the paper. I would love to take any questions and meet you later today at the poster session, where I also have some unpublished results. Thank you.
Any questions for Irene here? I have a lot of questions, but I don't know where to start. So let me recompose my thinking. So my understanding is, okay, first of all, I think this to me, I think this is going to be is very useful, not only for this setting, but I think this is applicable to gene expression also.
This is specific for methylation data.
Yes.
There is some beautiful work out there that is unfortunately not mine, but infers DNA methylation from gene expression.
Okay.
So from bulk RNA-seq. Yeah. And I think maybe from single-cell RNA-seq, but I don't swear. So if you don't have the DNA methylation data, you can still use it a little bit.
It's not an algorithm. So the assumption is actually you need a longer rate that you can so you can put this longer are for individual cells. That was the basis of the algorithms,
right?
Which used, and you try to deconvolute the signal, and the longer the rate, the more information that comes from a particular cell. Is that the basis of the algorithms?
Yes. So if you had single cell DNA methylation data, you would not have any problems. But the signal, the change between cell types in DNA methylation is very, very strong. While the change that is associated with disease can be very mild. So if you look at EWAS studies, so epigenome-wide association studies, a change of maybe 10% in methylation will be a huge change. While between cell types, like an eye cell and an ear cell, a change of 100% is likely. So even if you have a tiny bit of contamination, and you're trying to compare, as I was in the beginning, I was trying to compare pancreatic islets between people with diabetes and healthy individuals. If I even have a tiny bit of exocrine pancreas, which if you have pancreatic islets, you will get exocrine pancreas, it will be impossible to tell apart if the changes that you see are due to cell composition or true disease-related association.
And one more question is, you did simulation, because you have data for individual cell types. So yes. I get you actually try real mixing the data, mixing the cells itself, and then do the deconvolution. Have you done that? And also I hear from that your presentation.
Okay, so I have simulation data and in silico mixtures. I did not try in vitro mixtures.
Yeah, remix and then deconvolute.
Yes, okay. Which are an amazing resource, but they require a wet lab, which unfortunately we do not have.
I see.
But I think this field is really lacking benchmarking data sets. So if anyone has a wet lab and is willing to mix some cells and upload that data with the true
I can give the cells. I grow cells in the lab, so I can give cells. So, yeah, Thanks, Irene,
would
for
be
the great
amazing.
talk. I think wonderful introduction to